of which exist yet, that were published in, well, they mentioned them in Mushrooms of the Redwood Coast. They haven't published them yet. And then here, this one I saw on Mount Rainier a couple months ago. This one is Chimonophyllum. And so a beautiful white mushroom that looks really blue with the ultraviolet light. And these glowed so brightly that I didn't even have to wait for complete darkness to take this photo. I took it about 4 p.m. So just being in the shadow was enough to be able to show this fluorescence. And then here's one I took up at Rand Workman's house on Whidbey Island. This is a Atheniella, and they're not particularly fluorescent, except for the rhizomorphs at the base of the stem glow really brightly in ultraviolet light. So that's pretty unique. I haven't seen any other ones where just the rhizomorphs glow. And then here's a really common foliota that is in the wood chips in the Bay Area. And this one, people call it Foliota spumosa, but the spores are too large for the spumosa group, so I don't have a name for it yet. But it's really fluorescent and really common, usually mistaken for a gymnopilus, but it's got a viscid cap. And for this photo, I just brought the mushrooms home and took the bathroom mirror into my room for a minute to take the photos. And then here is Cortinarius sideliae. And this is a really viscid Cortinarius, a really slimy one. So it looks kind of like a tiger's eye with the flash and kind of greenish and ultraviolet. You can see I have the same photos here with regular light and ultraviolet light. And so what I do is I set up a tripod and then use the flash to take one picture. This one looks like I used an LED headlamp probably. And then just take the same picture from the same spot with the ultraviolet light. And that way you can see which parts of the mushrooms are most fluorescent and see how they change with the ultraviolet. This is Phomulina alnicola from Salt Point State Park. So these are usually misidentified for Hyphiloma fasciculare because they look really similar, but the spores are more rusty brown than the dark purple brown of Hyphiloma. And this is another one that I took during the daytime. There was actually pretty good light, but the ultraviolet light I used was really bright. So compared to the waning daylight, the ultraviolet light was much brighter, and you can't really tell that it's not taken at night. Here's one that I found in Michigan. This is a, we're calling it Foliota condensa. It's probably a different Foliota, really. But really cool, really bright ultraviolet light, ultraviolet fluorescent mushroom. And this one we found at Midwest Mushroom Camp. And we went on a night hike. And Elizabeth had brought this Convoy C8, which was a really bright 365 nanometer ultraviolet light. And, you know, on this hike, about 20 people had black lights, but this Convoy just blew all of them out of the water. So I immediately bought one. This one has an extremely bright LED, and it also has a filter that blocks the visible light. So the 365 nanometer LEDs also make a little bit of visible light in the green section of the spectrum. And then the filter blocks that, so you just get ultraviolet coming out. So it's a very pure light. And being that it's 365 nanometer, it's completely invisible to the eye. So you only see the fluorescence. So if you're going to get serious about ultraviolet photography, I definitely recommend a 365 nanometer light with the filter that blocks the visible light that comes out from the front of it. And the Convoy is extremely focused, so it's really good for finding things that are really far away. It's not as good for cell phone photography because the spot is so small. But what I do here is I just set up the tripod and I think this was like a 6-second exposure, and then wave the light around so it's kind of equal on all the different parts for 6 seconds. And then in this one, I just used the flash and flashed it about a dozen times from different places. And then this one, Sasha Ospina found, and these are really mysterious. What you're looking at here is a leaf. And as you can see, there's some brown spots on the leaf that don't look like very much. But with the 365 nanometer light, they're extremely bright orange. So I did some microscope work to figure out what these are, and I still have not, don't have any clue what it is. I just saw regular leaf stuff. So maybe it's bacteria. Some people say lichen, but I don't think so. But I've also seen this in Washington State, these really bright dots on decaying leaves. And here's another one that I found at Midwest Mushroom Camp in Michigan. And I was pretty excited when I found this Ganoderma applanatum because I'd never seen a fluorescent I'd never seen a fluorescent Ganoderma, and even more importantly, I'd never seen a mushroom that fluoresced red. But then when I looked at the photos, I realized that the mushrooms were not fluorescent at all. It was actually algae. So you can see this, the parts here that are green are red in the ultraviolet photo. So it turns out that chlorophyll fluoresces red. So a lot of plants, like especially poison ivy and mosses, look bright red. And oh, someone's calling me. Try to fix that one.
The wonders of integrated technology.
That was Joey. He probably wanted to do a cameo on the show. So I will call him back.
This has never happened before. This is awesome. First time for everything.
Right?
Don't dox anybody.
We all have Joey's phone number now.
Hi, Joey. You're on the live stream for the Central Texas Mycology Society. Yeah.
We cannot hear
people were asking on the YouTube if you were going to make a cameo on this live stream. So
I cannot understand what he's saying at all. I wish it were working better.
I detect a little bit of that Chicago accent.
This is working awesome. Yeah, I'm not hearing anything he's saying.
oh, damn. Well, jelly fungus usually is not poisonous, so you'd probably be okay. But there's a Facebook group, Poisons Help. So usually people post to the Poisons Help group, and there's like 100 people on there that identify stuff real fast. But after the live stream, if you want to send me photos of it.
Sounds like a bit of a something going on.
It's crazy. Well, I'm going to get back to my talk, Joey. It's good to talk. Okay. All right. No, I don't. It sounds very sketchy. Yeah, I have not seen that. It sounds extremely sketchy. I advise you not to buy them.
Yeah, this moment is truly unforgettable. The YouTube is saying.
I'll look at it, but I guarantee you I'll think they're sketchy. But I'm going to get back to my this live stream. I'll talk to you later on. All right, talk to you. Bye. All right. Close this.
Too bad that didn't work out better. We
Right?
just couldn't hear him.
Oh, you couldn't hear him.
No, so it was just a
Oh, damn.
one-sided conversation.
Oh, that's too bad. I've never done a live stream with a messenger call. Anyways.
That just one of many thousand of IDs that you help out with a year. I saw your stats on iNaturalist.
Oh, yeah. Yeah, I think I'm on track to do at least twice as many this year.
Wow, what was the amount? What was the number? Can you share with everyone?
It was 44,000 mushrooms that I identified for other people on iNaturalist in 2020.
It's incredible.
It's a lot, but it's really just a few minutes a day. So here's the Scanoderma with our bright red algae. And then here is a Gymnopilus. And so this one just kind of an old dried up Gymnopilus, but it's got a really cool fluorescent chylocystia. So those are the little cells on the edges of the gills. And here's a Hydnum, the little hedgehog mushroom. And you can see they're real purple, and that's partially because they look purple and ultraviolet, but also because they I used the 395 nanometer ultraviolet light for this. So most of the cheap black lights in the market are this 395 wavelength, and the 395 is almost invisible. You can just barely see it. It looks very purple. So in your photos, you get a lot of this purple color. So the 365 is a lot better. This is a hemimycena from the from Humboldt redwoods, and so a pretty white mushroom. They look really blue with the ultraviolet. And then here's the brightest fluorescent mushroom. This is Hypholoma fasciculare, and these are really common all over the world. And you can see another one of these leaves back here with the orange spots on them, the mysterious orange spots. So these ones are pretty bitter, kind of poisonous, but it's the most common fluorescent mushroom that you'll see. And all the parts of the mushroom are fluorescent, you know, the gills, the top, the stem. And one time I took this mushroom and I dissolved the pigments in water and a protein gel to see what molecule it could possibly be that's fluorescent. And I found that it's a really tiny molecule. So it's not a large protein or anything. It's a really small molecule that is fluorescent. Here's another Hypholoma. This one is Hypholoma lateridium. And Hypholoma lateridium is an edible relative of Hypholoma fasciculare. This is one from Michigan. And turns out that all the Hypholomas that I've checked are very fluorescent. Here's a Septitroma. So this one is from Texas on Big Thicket. And Septitroma is pretty neat because you see this neat purple color, which is not a real common color to see in fluorescent mushrooms. And here's another really mysterious one, also from Big Thicket. And this one is really bright with the ultraviolet light, and nobody knows what it is. We've got DNA sequences of it, and it doesn't really match anything. It's sort of close to glycoside, but really pretty far from everything else. And then a lot of Russellas are fluorescent. So this is a Russella from Gainesville, Florida, and they show up really well. This is one that I found just a couple weeks ago at Prairie Creek Redwood State Park in Humboldt County. And this one was under spruce. And Russellas are really hard to identify. So I sequenced the DNA of this one. And I was out on the road, so I actually set up a little lab in my car and ran PCR in the trunk, and totally worked. And I found out that this one is Russula zelleri. Here's another little brown Russula from Gainesville. And then lycoperdon. Most of the lycoperdons are not fluorescent, but a few of them are. This one didn't glow too much on the outside. Instead, lycoperdon under them, but the inside was glowing bright yellow and fluorescent in ultraviolet light. And then here's some sort of hydroxylaceae. So this one isn't really that fluorescent, but it reflects the violet very well, so it looks real purple in the 395 nanometer ultraviolet light. And there's a lot of fluorescent insects and animals as well. Recently in the news, they found that the platypus was fluorescent. And I discovered the other day that dogs are fluorescent. There's quite a few. But this is a little millipede from Texas. And you can see it looks really cool. The legs are just brightly fluorescent. Here's a fluorescent cactus from Arizona. And so this one here is just with flash around midnight, and then with 365 nanometer ultraviolet light, it looks like this. And then with and then if I put this is a 395 ultraviolet light. Then if I put an orange filter over the camera, then it blocks all that blue. So this is with the orange filter, without the orange filter, and with flash. With the 365, you don't really need the orange filter, but the 395 makes the orange filter makes the photos look a lot more dramatic. Here's a Centauroides, a bark scorpion. So it's one of the more dangerous scorpions. This is also from Tucson, Arizona. So these things are extremely fluorescent because they have beta carbolines in the shell. So this is 395 nanometer light, and then with the orange filter, cuts out all that blue, so you just see the fluorescence. And then here's some club moss. This is kind of cool because it looks real pink. This is also from Big Thicket, Texas. And there's a lot of fluorescent lichens, thousands of them. Here's one of my favorites, it's Achirospora socialis. And so with 395 light, and then with the orange filter, you can't see the rock at all. And one more fluorescent lichen, this is Spherosporus tuckermanii, and this one I found a couple weeks ago in the, it's the Oregon Coast Redwood. So this is the furthest north that you can find redwoods. And so it's this green lichen that looks really cool. I took this photo right around sunset, and I took this photo with a new lens I got. It's the Laowa 15 millimeter macro lens. So it's a macro lens that's also extremely wide angle. So I was able to get this detailed picture of the lichen, but at the same time get the sunset in the background using this really unique lens. There's more Spherosporus tuckermanii. And then this one is a bacteria. It was growing in a polypore over at Mount Rainier. Larry Evans found this. And they're more interested in the polypore, but when I pulled the black light out, those pink spots started glowing really brightly. So it turns out there's a lot of fluorescent bacteria. So the ultraviolet photography I've been doing for a couple of years now, and share a couple tips to make your ultraviolet photos turn out really good. You don't even need a fancy camera for ultraviolet photography. My cell phone works just fine, and the photos turn out almost as well. One thing you can do with the fancy camera is to take the exact same picture with flash and with the ultraviolet, but you could do that with a cell phone tripod as well. So for ultraviolet, looking for fluorescent mushrooms, you definitely want a 365 nanometer light. The 395 nanometer are the really common purple ones that are super cheap. The 365 nanometers are not much more expensive, though the really good ones are a little more. All the really professional ultraviolet photographers use a Convoy C8 or a Convoy S2 or something like that. And these are about $80 online. Then the Lone Fire SVO03 has a wider beam, so it's better for cell phone photography. And those are about $30 online. The Convoy is a lot brighter, so definitely recommend that, especially if you're looking for stuff that's really far away. And so that is all I have for fluorescent. So the other kind of glowing mushroom are bioluminescent mushrooms. And bioluminescent mushrooms make their own light. So when you're walking at night through the woods, you'll see these glowing things. And if you shine light on them, you can see them, but it doesn't charge them up or anything. They just, you know, they're just bioluminescent. They're not phosphorescent like some of the other like rocks and things. Actually, the lichens are slightly phosphorescent. If you shine a bright light on a lichen, they'll glow for a couple seconds. And a lot of plants too, very slightly. It's hard to see. In any case, the bioluminescent things are much more dim than the fluorescent things because you can use a really bright black light and make it as bright as you want. But for bioluminescent stuff, they're making their own light. So it has to be really dark out when you find them. So it helps to be really far away from a big city, though if you need to look near a big city, if you do it on a clear night, all of the light will go straight up into the air and it won't bounce back down. But on a cloudy night, it's just too bright to see anything near a city. So to see these things, usually you have to let your eyes adjust to the dark. After about 4 minutes, my eyes seem to be completely adjusted. The scientists say you need to wait 20 minutes, but I don't find that to be the case. Usually I start seeing them after about 2 minutes. And if there's any moon at all, then everything in the forest looks like it's glowing. Anything that's light colored, all the trash or white rocks or light colored plants look like they're glowing. So you want to go at a time when there's no moon. And to do that, you either wait until there's a new moon, but that's only once a month. So it's better to check the moonrise time and the moon set time. And to do that, I use an app that's free for Android called Sun Locator Lite. And there's some for iPhone too, or you can just Google it, moonrise and moon set time. But a lot of times the moon doesn't rise until 3 in the morning, or the moon will set at 4 a.m. So you can get up real early and have a couple hours of pitch darkness to look in the forest before it gets light. And then another thing you can look at is what's called astronomical dusk. And that's about an hour after sunset. Sun Locator Light or Google will also tell you when that is. And that's when the sun is far enough away down below the horizon that it's completely dark. So usually you want to wait till astronomical dusk before you go out and start looking for your glowing mushrooms. Though the fluorescent ones you can do way before that. Another cool thing you can do with Sun Locator Light is like when you're camping, you can see like exactly when the sun comes up. So you can like see, find out where to put your tent to be camping in the shade. And when you're looking for mushrooms that glow in the dark, you have to walk with no light at all. And that means that's usually sort of dangerous. So it's a good idea to wear glasses. That way you don't poke your eyes out, and they also maybe make your vision a little bit sharper so you can see stuff a little bit better. But also scope out a route during the day so you can walk, maybe find a paved trail through the forest. That way you don't walk off a cliff or into a hole when you're looking for mushrooms at night. Also, a lot of people use red lights at night, but I really prefer a white light at night, but you just have it super dim. So a super dim white light will not destroy your night vision because it's so dim that your eyes don't adjust to it. But you can still see pretty well when your eyes are adjusted really well to the dark. They don't really sell LED lights that are dim enough, but you can use like the display on your cell phone or just some other extremely dim white light to be able to walk at night through the woods. And when you're photographing bioluminescent mushrooms, they're so dim that you almost always need a long exposure. So you need a tripod for that. Some cell phones have a night sight feature, and that does work. But usually you need a DSLR and a tripod. And depending on how bright the mushroom is how long you need to leave the shutter open. So the brightest mushrooms that I have found, the brightest glowing mushrooms are Mycena curli, and those are down in Veracruz and Hidalgo in southern Mexico. And those are so bright that just do just a 6-second exposure. But then other ones are extremely dim, and I'll leave the shutter open a whole lot longer. So the more you turn up the ISO, the grainier your photo is. So you want to use as low of an ISO as you can, but also keeping your shutter open a really long time makes the photos more grainy. So there's a bit of a sweet spot there, and I find it's usually between 2 and 5 minutes. And then the aperture, if you open up the aperture all the way, you get much less depth of field, but you get a lot more light in. So you usually want the aperture open all the way when you're taking bioluminescent photos. And then you can use focus stacking to stack several photos which are focused slightly different places, so you can get the whole subject in focus at once. You can also do the kind of focus stacking where instead of increasing the depth of field, you take the same picture over and over, but it subtracts the noise from all of them, so you're getting a photo with less noise. It also helps to have a low noise camera. If you spend $3,000 on a camera body, it'll still take just about as good of pictures as a $500 camera body, but what you really get for all that money is a sensor that is very low in noise. So you'll be able to shoot at higher ISO and not get so much digital noise. So there's 2 ways I look for glowing mushrooms. One of them is to just walk at night with no light and look for stuff that glows. And the other way is to walk during the day and just pick every mushroom I see that might glow and put it in a talco box. And then when the sun goes down, or go to a dark room and take a long exposure of the talco box and see which compartment glows. They both work really well. Most of the glowing mushrooms are mycena. So if you pick everything that looks like a mycena, it's pretty rare that I pick a whole bunch of mycenas and at least one of them doesn't glow. So here's a mycena that I found in Gainesville, Florida. And this mycena does not glow at all, but the mycelium glows pretty brightly. So you can see all of the leaves here are glowing pretty well. And here's a close-up on a grape leaf that's colonized with this glowing mycelium. And you can see looks really cool with the glowing isomorphs. And here's one from Oaxaca. This one I found at about 3 in the morning. And this is Mycena lucifera. And lucifera comes from Lucifer. It means light bringer. So this was one that glows pretty brightly. You can see this from about 100 meters away. And sometimes you just find trunks that are just covered in this. And this photo here is 2 pictures. So I took one picture that was focused on the left, one picture that was focused on the right, both of them 90-second exposures, and then combined them in Photoshop to make one picture where it's all in focus. This one is Mycena lux follicula, and follicula means leaf loving. So this is a glowing one that grows on leaves. And I took this picture in Veracruz. I've also seen it in Jalisco. And in this one, the stem is the part that glows the brightest. You can see the leaves are also glowing, and as is the cap, but the stem is really bright. So when you find these, it usually looks like a vertical line in the forest that's glowing. This one is Mycena globulispera, and this one was recently described from Brazil. And the cool thing about this one is that the pilocystidia, or the gill edges, glow. And then the mycelium glows a little bit. This one does not glow very brightly. So this here is a 40-minute exposure. So I just turned down the camera and went to sleep. And after 40 minutes, it ran out of battery, and it turned out pretty well. But you can see it with the naked eye. When you find these things, you can see that they're glowing, but it's just really difficult. Here is one called Mycena fulguris, also from Veracruz. And on this one, only the stem glows. You can see in a lot of these, I take them home and photograph them. It's a lot easier that way. This one is Mycena nebula. And this is growing on liquid amber trunks in the cloud forests of Veracruz. And it's named Mycena nebula because sometimes there's hundreds of them on one tree trunk. So it looks almost like a nebula of stars with all these glowing mushrooms. And this is the first glowing mushroom I ever saw back in 2012. Here's one that doesn't have a name yet, but just the base of the mushroom glows. So this photo I took in the moonlight, so the moon was lighting up the caps. You can see the base is glowing pretty well. And then also where an insect took a bite out of the cap, that part also glows. And you'll notice some other ones do that, like Mycena haematopus is one that glows, but only where the insects bite it and the base of the stem. Here is Mycena stylobates. This one does not glow, but makes the leaf glow very well. So when you look at this, you can see the parts of the leaf that are being eaten by the mycelium, the lighter color of the mycelium bleaching it out. And that's the parts of the leaf that are being eaten and the parts of the leaf that are glowing. Here's one that's not named yet, but it will be named Mycena luxminer, and it is growing on a liquid amber seed pod. And so these mushrooms don't glow at all, but the mycelium glows and also the primordia glows. So the bright spots you see here are all the primordia that are forming in just about the fruit. This one sometimes is really prolific, especially in Tamaulipas. There's a place called El Cielo. And up there, there's just carpets of the leaves that are covered in this. And just everywhere you look in the forest, there's just thousands of leaves in your field of view that are glowing. This is Mycena perlae, which is the brightest glowing mushroom that I have seen. The scientists say the brightest glowing mushroom is Neonothopanus gardneri from Brazil, though they haven't compared it to this one. So Mycena perlae is one that I first saw in the state of Hidalgo in 2014. And so in this one, only the cap glows. And it's so bright that if you pick it and you throw it, it looks like a shooting star. Here's one where I lit up the stem with a little bit of light from a headlamp or flash. So when I first found these, I told my friend that I was going to go look for bioluminescent mushrooms, and it was 2 a.m. and raining, and he just laughed and said, "Oh, have fun." But after I walked about 100 yards and stepped in a few puddles, all these glowing mushrooms, and, it turned out to be a new species. So these, sometimes there's thousands of them growing in pine needles, occasionally oak leaves. So they can look like stars all over the forest floor. There's a place near Jalapa that I go where there's just tons and tons of them. Here's one that's not named yet, but it will be named Mycena lux pellicula. The Lux pellicula, and it's named that because these mushrooms have a separable gelatinous pellicle that you can peel off of the cap. And pellicula means film in Spanish. So the Mycena pelliculosa will have a cap that's peelable. These are always growing on the leaves of the rubiaceae. Here's a really interesting one, probably a resin of Mycena. So the mushrooms don't glow at all, but it makes the stick glow pretty brightly. And this one I found in Jalisco. And then here's a really interesting one that I also found in Jalisco. This one I have no idea what it is because there weren't any mushrooms, but the mycelium was glowing pretty brightly. So these isomorphs just kind of fly through the air, and these were several feet off the ground in these sticks that were hanging in the air. So it's glowing mycelium. So here's a little bit about the light spectrum. Ultraviolet is the most dangerous type of light, and it's over here on the left, the shorter wavelengths. And then as the ultraviolet wavelengths get longer, it gets less and less dangerous. And then right here is about 400 nanometers, and that's where you can start to see it. So the ultraviolet lights that we usually use are about here, 365, 395. So they may be a little bit dangerous. Some people wear eye protection when they use them. I heard that you can get cataracts from them, so permanent eye damage. I don't know if that's true, but they're not nearly as dangerous as the ultraviolet lights that are over here. I got one recently that's a 254 nanometer. So it causes sunburn, causes severe eye damage, and it causes skin cancer. So when you use those, you have to put on sunscreen and wear glasses. And then over here, you have the visible light, and right here is 535 nanometers, and that is the color of all of the mushrooms that glow in the dark. So all of the glow-in-the-dark mushrooms are just that one color, whereas the fluorescent mushrooms can be a whole lot of different colors. And then you can see up to about 800 right here, which is the infrared. And so one thing I wanted to show you really quick is with iNaturalist, when I find fluorescent mushrooms, I have a project on there. Let's see if I can just. Turn it up with Google. Yeah, so there's a project called UV fluorescent organisms on iNaturalist. And so if you find anything at all that glows in the dark, you can join this project. And then when you take pictures of it, you can tag it either in the app or here in the website. And you can, then it kind of like keeps track of everything that's fluorescent. So everybody in this project together has found 353 different species that are fluorescent. And they've got 108 people that have contributed to it. That's probably less than 1% of the mushrooms that are fluorescent. Every time I go out with black lights at night, I found a new mushroom that I didn't know was fluorescent. Most recently, I found Rhodophyta nidulina, and that one was extremely fluorescent, a funny little mushroom that I see in Mexico and on the West Coast. It smells really weird like burnt rubber, and it glows really brightly. And over here, you can see Hypholoma fasciculare is the most commonly observed fluorescent thing. But this scorpion, that's our West Coast scorpion here, is really fluorescent. Here's a really cool one, Zistochira dissecta. This is one I see a lot. And this one, it looks like this. Let's see if I find pictures of it glowing. Yeah, here's one. It looks like one of my pictures. These glow really brightly. So the really fun thing to do with these, yeah, these are the ones that I took just the past few days. Yeah, they glow really brightly in ultraviolet light. So the fun thing to do is just take a video on your phone. Oh, that was not on. So if you take a video on your phone, they look really cool, just with the legs being so fluorescent. And is the range of that. So one more resource, and then I'll open it up for questions, is that on Facebook, we have this fluorescent photography forum, and it's called Fluorescent Forests. So if you just search for that, there's a lot of really cool stuff on there. Here's the talk, but there's a bunch of different ultraviolet photographers on here. So they're doing ultraviolet stuff and also bioluminescent stuff. And maybe if I can scroll down to this this one rose that got posted yesterday. There's this one guy that posts just amazing stuff. There's my my poison hemlock. Looks kind of cool in black light. So here's a slime mold that has a really fluorescent plasmodium. We haven't figured out what that is. But here's this rose. So some of these plants can look really cool. And so if you're interested in fluorescent photography or bioluminescent stuff, that's a good Facebook group to join. So if anyone has any questions, I can answer them now.
Alan, can you tell everyone about the new species that you all found in, I think you said near Jalapa in Veracruz, and kind of the process too of like how you go about, identifying it, verifying it, determining it's a new species, and then how you go about the process of putting it into the taxonomy.
Let's see. Let's see if I can find that paper. So I found a whole lot of mushrooms. Let's see. Here it is. A whole lot of glowing mushrooms near Jalapa. So here's the paper that published with a bunch of people. But these, I used to say that Brazil had the most glowing mushrooms of anywhere in the world because I found like 19 glowing mushrooms in Brazil. But I have seen 26 species of glowing mushrooms in the Jalapa area. So many of them are extremely rare. So what I was doing is just collecting these mushrooms that were glowing. And after a while, we got a bunch of collections and published this paper that named 6 of them. And so these are all, they're all made with microscopic descriptions, macroscopic descriptions, and DNA sequencing of the ITS gene. And then I think, with this sort of publishing, usually you have to pay the publisher instead of the authors getting paid. So I think I paid about $300 to get this article published, which is standard in scientific publishing. They wanted like $2,500 to make it open access, so they didn't charge people $35 to read it. But there's a lot of undiscovered glowing mushrooms out there. I think maybe just about 10% of them are discovered so far.
I have a question. What makes the fungi bioluminesce? Like, how do they do that?
The bioluminescent fungi have an enzyme, and the enzyme is called luciferase, and it interacts with a chemical called luciferin. And when the luciferin and luciferase react with oxygen, it releases a photon. So chemical reaction very similar to a firefly, but they're different luciferins and luciferases than are in the animal kingdom.
Are there any hypotheses to why they bioluminesce?
No one really knows, but most people guess is that it's to attract insects. Because if insects can be attracted to fungi, then they eat them, and that spreads the spores and mycelium around. So fungi benefit a lot from being eaten. And that's why the poisonous fungi are just sort of random. They're not like meant to ward off predators or anything. Just random chemicals that happen to be poisonous. But I think they're, all of the mushrooms that are bioluminescent are these kind of little wood-loving saprophytic things. So I think they use the insects to carry the spores and mycelium around the forest and get a head start over the other mushrooms that have to wait for someone to wait for the spores to fly through the air on a wind current or something like that.
Is there anything that make mushrooms more likely to be bioluminescent than other mushrooms or even fluorescent?
With the fluorescence, I haven't noticed any rhyme or reason to it. So it seems like, mushrooms just have so many random chemicals, and many of them are not known to science yet. And a lot of chemicals are fluorescent. So it's just kind of a reflection of the chemistry of a mushroom, which colors it fluoresce. With the bioluminescent, most of the bioluminescent ones are in the genus Mycena, but there's also some in Neonothopanus and Pinellus, Omphaliotis, Armillaria, Geronema, and a couple other genera. But they're mostly related to Mycena. But there's really nothing you can do to predict that a mushroom is bioluminescent. You just have to know which ones are bioluminescent. Or if you know that lots of mycenas are bioluminescent, then you can just collect anything that looks like it could be mycena and see if it glows later on.
Are there any that people cultivate for their bioluminescence, or is there
There's
anything
two
that
that
goes on
people
for that?
grow commonly for bioluminescence. There's Pinellus diptychus, which is the most common one. That one's really closely related to mycena, even though it's kind of like a polypore. So that's why it glows. Some people even say that it should be a mycena. And that one you can order spores online from Sporeworks, or people trade around culture dishes and stuff. It's also really common all over the Midwest and West Coast. People say the West Coast varieties don't glow, but I have photos of that glowing as well. The other one that people grow that is bioluminescent is the Omphaliotis nidiformis from Australia. That one looks a lot like an oyster mushroom in that it's white and grows on wood, but it's bioluminescent and also pretty poisonous. So people don't grow that nearly as much. It would be kind of annoying if it escaped into the United States, because people in the United States consider oyster mushroom looking things to be safe to eat. Some that are not available, but would be really cool if they were, are some of these mycenas and the Neonothopanus gardneri. The mycenas I've spore printed and tried to cultivate, but the spores just didn't germinate. So I might have to clone a fresh mushroom directly to agar to get a culture of some of those. The Neonothopanus grows in Brazil, so that somebody down there would have to find that and culture it. But it has been cultivated in the lab in Brazil. It's just the culture is not available. Yeah, you definitely can grow these things. I don't think there's any mycorrhizal mushrooms that are bioluminescent. So that means that every glowing mushroom can, in theory, be cultivated.
Very cool. Let's see, trying to pick out some more questions from the chat here. Let's see. I guess we might have covered this already, but maybe for some more clarification, is it the same reaction for bioluminescence and UV induced fluorescence?
No, it's different. The UV is just taking a shorter wavelength with more energy and emitting a longer wavelength with less energy. So it basically saps some of the energy from the ultraviolet light, and that makes it visible. Whereas the fluorescence is like a chemical reaction that's making its own light. There's only one mushroom that I've seen that's both fluorescent and bioluminescent, and that's a mycena called Mycena quinaltensis that I found in Oregon. And that one, the mushroom glows in the dark and it glows in the ultraviolet. So that one is pretty cool.
Let's see. Do we find common secondary metabolites across bioluminescent organisms? Is that something you would know?
That's a good question, but I don't know. There's a lot of different metabolites and stuff, and it would be really cool to be able to do some of this analytical chemistry stuff. I'm not set up for analytical chemistry, so I don't know, but I know there's a research team in Brazil and another research team in Russia that's studying this stuff, so those guys might know.
Do bioluminescents fade after you pick them, and how long does it take if it happens?
Yeah, the bioluminescence fades, and by the time that the mushroom fully dries out, it's faded completely. So how long it glows is pretty much just how long the mushroom stays alive. So it'll stop glowing if it gets eaten by bacteria or if it dries out. So if you keep them wet, they can glow for a few days. But a lot of these mushrooms are really tiny. So as soon as you take it out of a moist tackle box, then it'll dry out right away and stop glowing within just a few minutes. Whereas a larger mushroom like an Omphaliotis or a Pinellus, they're fleshier. They don't dry out quite as quickly, so they'll last a lot longer before they dry out. But there's no mushrooms that continue to bioluminesce once they're dry. Though there are mushrooms that are fluorescent when they're dry.
So, like, if you wanted to make it last a little bit longer, could you give it a little bit of water or something?
Yeah, definitely. A lot of times what I'll do is just keep them sealed up in the tackle box. And if it's something real small, I'll put a little bit of moss in each segment of my tackle box. And that way That way they don't dry out until I take them out. But yeah, anything you can do to increase the humidity, even a humidifier, will give you more time to take pictures of them before they dry out and stop glowing.
Cool. Are the spores bioluminescent or fluorescent? Have you ever looked into that at all?
I have not seen any fluorescent spores or bioluminescent spores, and I don't think any of them are. A lot of mushrooms have very bioluminescent gills, but when you're looking for them at night, when you look under the mushroom, you see the spore print there with white light, but you can't see the spore print really at all with the ultraviolet light. So the spores are not fluorescing. Another thing I'll do is just take a little bit of a fluorescent mushroom and put it in the microscope, and then shine the Convoy C8 at the microscope slide from the side, and then you can see which cells in the mushroom are actually fluorescent. And I've never seen spores fluorescing that way, but like in Hyphiloma marginatum, I see that they have this chrysocystidia that's fluorescent. So it's just certain cells in the gills are brightly fluorescent. Different fluorescent species have different parts of the mushroom that's fluorescent.
Are there any is there any research into like the genomics in bioluminescence? I guess we kind of maybe already talked about it a little bit, and any study into the genes responsible for bioluminescence and fluorescence?
Yeah, there's two teams doing the full genome sequencing on bioluminescent mushrooms, one in Russia and one in Brazil, and they've published some papers, so they have figured out how that pathway works and which chemicals it's making exactly. As for fluorescence, there's a lot less research on that. I don't think anyone's done any full genome sequencing specifically targeted at fluorescent mushrooms, though I'm sure if we look through all the mushrooms that have full genome sequencing, then full genome sequences available, you could find some that are fluorescent. But it would be really cool to do full genome sequencing on a fluorescent mushroom and also its closest relative that's not fluorescent to try to figure out which gene pathways are making the actual fluorescent compounds.
Yeah, that is really cool. Lots of inspiration for any young students out there who want a thesis project. Do you know anyone who's researching any of the applications to bioluminescence? I know you just kind of mentioned those teams that are working on it.
A lot of people want to use bioluminescence to replace lights. It's probably not very practical because the mushrooms only glow when they are fruiting, and they're not very bright, even in the brightest ones. And maybe they can be made a little bit brighter, but still, they're not there all the time. I have used like a bag of Pinella stypticus mycelium for a nightlight, and it glowed for about 18 months. But it was very dim. So like if I woke up in the middle of the night, I could see this glowing bag of mycelium, but you definitely couldn't use it to find your way around or anything. But yeah, a lot of people want to use this chemical reaction to make light. Though I think ultimately LEDs will turn out to be a more efficient and more practical way to make light.
Yeah, that makes sense to me too. They're just so dim, like you were talking about earlier with the camera, that you have to have such a long exposure just to get like a vibrant picture, and it's kind of going the opposite direction with light generation there. Somebody is wanting to know, and a couple people asked, like, how can they contribute to this kind of work, just as like as a I don't know, citizen scientist. And
Well, especially with fluorescent, but also bioluminescent mushrooms, there's a lot that is not known. I would say that most of the fluorescent mushrooms, nobody knows they're fluorescent. And so if you can just bring a black light out at night and walk around and record which mushrooms are glowing and upload them to Mushroom Observer or iNaturalist, that's a good thing to do. On iNaturalist, they have that project that you can join. There's one for bioluminescent organisms and another for fluorescent organisms. And that's a good way to keep track of what's fluorescent.
So here's one that I think is kind of interesting. I know we talked about the bioluminescence being created through like a chemical reaction from an enzyme and a yeah. it's created through a chemical reaction, but are there any of the fluorescence created by bacteria in lichen and fungi, or is that something that's not really understood yet?
That's a good question. You know, I'm not sure what's fluorescent in lichens. My guess is it's the fungi, but you know, there can be fluorescence in the algae too. Um, but I think most of the weird chemicals in lichens are being created by the fungi. So I think it's the fungi that are creating these fluorescent compounds. But you know, fluorescent lichens can be extremely beautiful. I noticed about a month ago, Steve Axford got on a fluorescent lichen photography kick and was posting some really amazing photos of fluorescent lichens from China.
Yeah, he's great. We had them on to talk a little while back. So if anybody hasn't checked that out, people should. Talking about their new movie. That's pretty amazing. Everyone should also check that out. Let's see. He already talked about the why they glow, the bioluminescent fungi glow the way they glow. Let's see. Do you have any knowledge of orline and fluorescence in Cortinarius? Or
You know, Orlanine is pretty rare in the United States. And when I first got black lights, I thought that I'd be finding a whole lot of fluorescent Cortinarius, but it turns out that most of the Cortinarius I find are not very fluorescent. And the few that are fluorescent are not too spectacular. There's probably a few out there that are fluorescent, you know, they're really cool, but I haven't been too impressed with Cortinarius as far as a genus of mushrooms to do fluorescent photography on. I know Damon Teague found one where it wasn't too fluorescent, but then when he put potassium hydroxide on it, it was brightly fluorescent just where he put the KOH. So that's really cool. But I have heard that orlanine is deadly and fluorescent, but I'm not sure if that's a reliable way to find the deadly Cortinarius or not. I've never found one of the Cortinarius to have the orlanine in them, so that's definitely a good thing to look at. So for the ultraviolet flashlight recommendations, I'm using usually I use a Convoy C8, and the Convoy C8 is extremely bright. It's a 365 nanometer flashlight that has the black filter in it. And those are, they take the lithium 18650 cells. And that's probably the best one. They're a little expensive because they're like $80, but they're easily worth it if you plan to be walking around the woods at night. And then the other one that's not nearly as bright but has a broader beam, so it's good for ultraviolet photography, is Alone Fire SV003. And there's a whole bunch of other ones that also work as long as it's a really bright 365 nanometer light that has that black filter on it that passes ultraviolet but blocks visible light. You know, a lot of different brands will work. UV Beast is another one. And then I see someone asking about the orange filter, and that you can buy orange filters for cameras. Though what I do is I have this orange piece of cellophane. It's made for doing gel electrophoresis, and the Odin sells it. So I just hold the cellophane over the camera lens, and that works pretty well. I don't use it when I have a 365 light because the 365 is already invisible. But you really any orange kind of translucent polycarbonate piece of plastic, whatever, will work pretty well for filtering out the purple. And orange isn't even the best thing to use. The best thing to use would be like some kind of bandpass filter that cuts out maybe passes like 430 nanometers up to 800, and then blocks anything below 430, and that would give you much better color in your photographs. You know, something that's as close as possible to just cutting out the wavelengths that your light is using would be the best.
Could you do any of that filtering in like Photoshop afterwards, or is it just better to do it on the original photo?
You can do it in Photoshop afterwards, but if any of the color channels get saturated, you start losing information. So if you're going to do that, it's good to underexpose or take several exposures with different exposure lengths. But yeah, you definitely can do that. Other thing I do in Photoshop is like when I take a picture of a glowing mushroom, a lot of times there's noise from the sensor because I'm using high ISO and long exposure. But all of the mushrooms glow just one color, that green. So you can turn down basically turn off the red channel and the blue channel, and that cuts out more than 2/3 of your noise. So that's helpful. And there's also like a bunch of algorithms for, fixing noisy photos.
Okay, cool. Are certain types of fungi more likely to bioluminesce, such as ectomycorrhizal like the Russula or the Mycena?
Well, I think this question means fluoresce, since the bioluminescent ones are the ones that are glowing. So the ectomycorrhizal ones, none of those glow in the dark. All of the bioluminescent mushrooms are they're sapotrophic. But as far as it seems like as far as fluorescent mushrooms go, it's really random which ones will glow and which ones will not. And it doesn't really matter if they're ectomycorrhizal or sapotrophic or anything like that. There's some genera that are more likely to fluoresce than others. Like more than half the russulas are pretty fluorescent. Whereas something like chanterelles or boletes very rarely fluoresce. But the question says, are there any types of fungi that are more likely to bioluminesce? And definitely things that are wood lovers are more likely to bioluminesce, so things that grow directly from wood. And things that are either in mycena or closely related to mycena are much more likely to glow in the dark than things that are not closely related to mycena.
And in Texas, we have the common one that we have is the jack-o'-lantern or the can't remember the Latin.
Yeah, Omphalota subaludens.
Yeah. And that's related to the one that you had mentioned from Australia, right? That we don't want to come over.
Yeah, in our California, Omphalotus glows as well. The Omphalotus olivascens. And then there's a blue Omphalotus in Mexico that does not glow. I see up here someone says, are you involved in Mushroom Observer beyond being one of the top contributors? And when is the MO app coming out? As far as Mushroom Observer goes, I help run the servers a little bit over there. So I don't really do coding on the site, but if the site crashes or there's some sort of security issue or some sort of problem, then I'll log into the servers and fix them. And then the MO app, nobody's working on one now, but it would be nice to have one. For a long time, I didn't really want there to be an MO app because the cell phone cameras were so terrible that just kind of flood the site with low quality observations. But now that cell phones can take really nice photos, I think it would be nice to have an app for that, for Mushroom Observer. So if anyone would like to create an app for iPhone or Android for Mushroom Observer, I can give you the API documentation, and that would be a good thing to have.
Go ahead.
A question here. Cris que haya relación entre bioluminescencia y el efecto inmunomodulador de algunos hongos. So they want to know, is there a relation with the bioluminescence and immune modulating properties? And I don't think so. None of the mushrooms that are bioluminescent are known to stimulate the immune system in any way. So I think those are completely different groups of chemicals that are doing that.
could you give us a rundown of your photo setup for, or the camera setup for taking those photos you shared with us earlier?
Yeah, let's see, where do I have? Well, let's see, I think I have it all right here in this backpack right with me. Um, so I use this is a Nikon Z7. So it's a full frame camera, and then this is a 105 millimeter Nikon macro lens. So this is not the best lens in the world for taking photos of glowing mushrooms, because the largest the aperture goes is f2.8, whereas if you some lenses go down to like f1 or even more. So those would be even better. But still, it's a really good lens for mushrooms. It gets really sharp photos, so I just have to leave the exposure open a little bit longer or turn the ISO up. So I use this camera, and then I have, a tripod. This is just a cheap Manfrotto tripod for keeping everything really still and doing the long exposures. And then I also have it's in here somewhere. Oh, somewhere there's a really tiny carbon fiber tripod, and those are really good for taking pictures of mushrooms that are right on the ground. So, a big tripod like that is great, but the lowest it can go is about 1 foot. So, like another tripod is really good for doing mushroom photography right on the ground. And then let's see what else I have. Here is this is a Laowa 15 millimeter macro lens. So this is a very wide angle lens that also can focus extremely close. So you can have the subject almost touching the lens here, which makes it look extremely large. But it's a very wide angle at the same time, which gives a pretty cool effect. And then, let's see, I also use, here's just like a regular Nikon speedlight type thing here. And so this flash is really good. And what I do is I set up the tripod, and then I just flash it maybe a dozen times in an arc over the lens. So I just kind of go like that. And that makes it so the light comes from a dozen different directions and gives you a really nice light. It looks sort of natural. You don't get that harsh shadow like you do when you use the flash that's attached to the camera. And also you can do like more flashes from above than from the side to make it look like the light's coming from above. Or you can flash it like 7 times on the right and once on the left to give a really, really dramatic lighting. So, any just little handheld flash will work for that. And, that's pretty much it. There's a few other things I think I see. Here's the Alone Fire SV003, which it says is 10 watts. It probably is not really 10 watts. But this, if you look in the end of this light here, you can see it looks black. And that's the filter that filters out the green light. So if you take the black filter out of this, or just use a really cheap 365 nanometer flashlight, you'll get this green light along with your ultraviolet light, and it'll kind of add an aspect to your photos that you usually don't want. So the filter is pretty key. But that's pretty much all I use to take pictures.
I feel like I saw you shared a few photos a while back with the Pixel 3. Do you use that out? You doing bioluminescence or fluorescent? Not bioluminescent, but I'm guessing the fluorescent.
Yeah, the Pixel 3 XL is the camera that I got, and I like it quite a bit. It's a pretty good phone, and the fluorescent photos are so bright because you use a bright black light that a cell phone has no problem taking pictures, of these glowing mushrooms. And For bioluminescent mushrooms, you can use the night sight feature, but it's still not nearly as good as an actual tripod and long exposure. But for really bright bioluminescent mushrooms, you can get real good pictures of them with these features. These cell phones where you have to hold it still for about 10 seconds, and then it kind of concatenates that all into one brighter photo.
Yeah.
But if you're going to do cell phone photography with ultraviolet lights, you need to get a light that is not such a little spot. And I don't know of any lights that are focusable that are ultraviolet, though there probably are some. That would be really nice. So you could have a beam to find stuff, and then when you actually find something, then expand it out so you have this broad beam of light. Because with something like a Convoy C8, you have a very focused beam of ultraviolet light, which is great. But then when you have the mushroom in your hand and you're trying to take a picture with your cell phone, you just have a spot where it's really bright from the flashlight. So for cell phone photography, you want a light that can have a really broad beam so it can illuminate everything at once. And then you can take really cool photos of ultraviolet stuff with the cell phone. And I don't even night sight feature for the ultraviolet photography. It's so bright. This fluorescent stuff is so bright that just the regular camera works fine for taking pictures of fluorescent mushrooms.
Do you know a little bit about the history of like how people learned that mushrooms were fluorescent under UV light?
I think Brian Perry has a talk which touches on this a little bit, and it's recorded. You can probably find it on YouTube, really good bioluminescent talk. But I think in that talk, he said something about how some guy like 2,000 years ago, maybe Socrates or someone, wrote about these glowing fungi. And then there's some other accounts of, from historical accounts from hundreds of years ago, people noticing these things. They weren't really studied scientifically till much more recently. But yeah, they've definitely have been noticing them for a long time. And I think, more recently, people are less likely to notice glowing mushrooms because now we have lights and we always use them when we're walking at night. But if you want to see, if you use, a headlamp or any kind of flashlight at all, you're not going to see the glowing mushrooms. So I think people used to be in contact with them a whole lot more often when they were doing a lot more walking at night without light.
Yeah, that makes sense to me. Let's see. Someone was asking about your microscopes in the back. Do you have any microscope recommendations for beginners?
Oh, yeah. So I got two types of microscopes here. This one here, I like quite a bit. This is an M scope, and this has a zoom, so it can zoom from 3 to 90 times, actually 7 to 45. So it's a stereoscope. So I use this microscope for actually just preparing the slides or looking at like ascomycetes and stuff. And then a compound microscope like this is what I actually use for looking at the mushrooms. And so this is a Nikon microscope. So it has a halogen light. It's really bright. It has some of these like high-end objectives on here. But for beginners, I usually recommend like something like an M scope. The M scope B120 is about $200 and takes really good pictures, has nice clear images, and is almost as good as this expensive Nikon here. So I think those are definitely a good one to get. There's also Omax, makes about the same scope for a couple $100. And then if you want to spend a little bit more, M-scope sells like plan achromatic objectives. So those are like the real expensive objectives that can be thousands of dollars each. But them for more like $20 to $200. So you can like upgrade an M-scope with these really fancy objectives, or get one of the higher end M-scopes that has these plan achromatic objectives already installed. And the optical quality on those things is really nice. But if you just want to do science, you don't really need an expensive microscope. I think for identifying mushrooms, a $200 microscope will work just as well. is a $20,000 microscope. But the more expensive microscopes are a little bit easier on your eyes, and they're better for photography, so you can take clearer photos. But for beginners, I think it's best to start out with like an M-scope B120 because they're cheap, they're easy to get, and they work really well. And if it turns out you really like microscopes, a lot, then you can upgrade to a fancier one. But microscopy is really hard. And I think most people that buy microscopes don't realize how difficult it is to get actual information out of a microscope. When I first bought my first microscope in 2007, like everything I did before like 2011 or 12 was almost worthless because I didn't know what I was supposed to be looking for. There's thousands of kind of jumbled shapes in there. You don't really know how to assign meaning to it. And what I should have done is just ask somebody that did this for a living, like, hey, how do I use my microscope to identify mushrooms? But instead, I was really stubborn, and I just studied on my own and sat down in front of the scope for hours every day, studied the keys, and I eventually learned it. But these days there's a lot more resources. There's a PDF for fungal microscopy. There's the fungal microscopy Facebook group. People can just ask me about stuff. And I'll be happy to teach people how to use the microscopes. And I've been making a few microscope videos as well. But microscopes are really cool because you see a whole other layer of beauty in the fungi, but they are not easy to use. It's even if you're really good at it, it's going to take some time before you can actually get any useful information out of a microscope.
We'd love to have you back to do a beginner class. I don't know how useful it would be over Zoom. Do you think it would be possible to do a class?
I think I could do a Zoom class, especially if everyone kind of has a microscope and they can kind of follow along or something like that. Though maybe that's a little difficult and it's better just to wait till COVID is done and we can do it in person. Yeah, I mean, I do like microscopy. It's really fun just to sit there for hours and hours and find out all this stuff. There's so many beautiful things you can see in the mushroom gills with these microscopes. Usually I teach the microscope classes in person.
Yeah. And we all love the meme of you that Crystal made.
Yeah.
That's pretty good. That's
been around. Crystal makes good memes.
Yeah.
Let's see. Which is your favorite of all the bioluminescent and fluorescent fungi that you found?
Let's see. The bioluminescent fungi, I suppose my favorite might be Mycena globulispora, because just the gill edges glow, and that one's really cool. I also found a glowing Mycena in California just a couple weeks ago, and I don't know which one it is yet, but most of the Mycenas in California, just the stem base glows, but this one, the whole mushroom is glowing and very brightly. That one was under Monterey cypress over in Alameda County. So that one was pretty cool to find. I suppose I like the ones where there's just they take over the whole forest floor and make the whole forest floor glow. And I've seen that in Veracruz and also Tamaulipas. So those are really neat because there can be 100,000 leaves within your field of view and every single one is glowing. So it's a pretty unforgettable place to camp. As far as the fluorescent ones glow, it's really hard to beat Hyphiloma fasciculare just because it's so bright and common, and a lot of times it's in really good shape. And even when there's barely any mushrooms out, you'll still find those in pretty good shape. And they're just brighter than really any other fluorescent mushroom. But the russulas can look really cool too. I suppose really my favorite is whichever one's in front of me.
That's such a mushroom lover's answer. My favorite one's the one that's in front of me. I love it. Can you briefly talk about the DNA sequencing at home, like your technique for doing that or doing it on the road?
Yeah. So what I do is I just take a little bit of mushroom, put it in a tube, and extract it with like sodium hydroxide and a couple other chemicals. And then I use a PCR machine to copy the DNA. Here's the PCR machine that I use here. This is a Biorad mycycler. These are pretty nice. You can see it's got 96 wells here, so you can do you can sequence 96 mushrooms at a time. And you know, this one I got a 2,000 watt inverter for my car, so I can run it in the car. But they take kind of a lot of electricity, so you got to keep the car running. So it's usually best just to dry the mushrooms and bring them home. But in any case, I put them in there and let that run for a couple hours, and then after that finishes, use a electrophoresis gel to see if the PCR reaction worked. And if it didn't, I rerun the PCR with a new DNA extraction. And if it does, then I send it off to either GeneWiz or Eurofins. And GeneWiz charges about $8 per reaction to do the sequencing, and Eurofins charges about $3.50, but you got to do 96 at a time. So either way, the price is really low compared to how cool the data is. And once I get the data back, then I test it and correct the errors in the chromatogram, upload it to iNaturalist, Mushroom Observer, and GenBank. So the DNA is always there for other people to study. But there's really no part of this that's particularly difficult to do at home. You know, you don't need like sterile conditions like you do for growing mushrooms. The chemicals are pretty cheap. The primers are cheap. Only the sequencing itself is a little bit expensive, but still, you know, maximum of $8 per sequence. That's still pretty cheap. And you know, usually I'll do like 24 mushrooms at a time, because even if I do one of them, it still takes me, you know, a few hours, and kind of still takes all day. If you do 24, it takes all day. You can do 96 in a day, but then you're really doing a lot of repetitive pipetting. So, you know, I like to do batches of 24, but there's, you know, the equipment's really small. It all fits within like one of those Tupperware tubs, and there's nothing that's hard to get. Only the DNA gel stain is a little bit toxic, and even that's not particularly toxic. So given how small it is and how available all of this stuff is, there's no reason you can't do it at home and get you know, the same results that you would get in the high-end laboratory.
Where do you get your reagents and how?
A lot of the reagents I get from a place called the Odin, and the Odin is a biohacker supply place. And so if you just go to like the-odin.com, they have the PCR master mix and stuff like that, the fungal primers and stuff like that. But I usually order primers from a company called IDT. And then the other chemicals, if the Odin doesn't sell it, then I'll just get it from eBay. And I think that's mostly the DNA extraction stuff. I use a DNA extraction protocol called Extractinamp, and it uses sodium hydroxide, tris, potassium chloride, and EDTA and BSA. So these are kind of like pretty standard chemicals that you can get on eBay. And you know, it's a little bit expensive if you buy the chemicals, but then you mix up enough to do like, you know, 100,000 mushrooms or something. So, you know, they're not too expensive in, you know, at scale. It's just that whenever you buy any chemical, even table salt, if you don't buy very much, it's going to be kind of expensive. That's just how chemicals work. But yeah, eBay and the Odin, between those two places, you can get everything that you need. And the Odin has all of the tubes and pipette tips and you know, everything else, all the consumables that you would need. Pipettes I use. Pipettes like these, you really only need probably 2 of them. The most important is the tiny pipette. This is like a 20 microliter pipette, or 10 is even better. And then here's a larger pipette just for like mixing up the master mix and stuff like that. But there's a Facebook group that I started called Fungal Sequencing. So if you're curious about sequencing stuff, you can join that Facebook group and ask questions about it. or you can just message me. I'm always happy to answer private messages about DNA sequencing or microscopy. They're a lot better than most of the stupid private messages that I'm answering all day.
Oh yeah, DMs, man, they can get pretty wild.
Yeah.
How do you fund your research, or how can people support your research?
Right now, I do a bunch of different things. Yesterday, I did a photography shoot for for a microgreens company. But usually I'll do like DNA sequencing for people, or I'll do lead mushroom hunts, sell photographs to book publishers, give talks, or various stuff like that. And if you like, before COVID, you could pretty much just sign up to give a talk every weekend, and then travel around and hunt mushrooms all the time, and that was pretty self-sufficient. These days, it's a little more difficult. But let's see. I think I do have a Patreon, but I haven't really used it in a while. I don't usually, ask for donations or anything. What I really need more than anything is time, because I have the microscopes and all the DNA sequencing stuff that I need. But maybe I'll do like a Kickstarter or GoFundMe or something to get like $100,000 microscope one day. But yeah, just all sorts of random stuff. I do have a Patreon, but I don't really use it.
Drop your Venmo right now. Someone was asking you to dish a little bit about the taxonomic upheavals. Like, what are your thoughts on that? Like changes in nomenclature, and it's a little bit off the bioluminescent topic, but I think it's really interesting, and would love to hear your thoughts on.
Yeah, so it used to be that nomenclature was based on what things looked like, how people thought they were related, based on the microscopy and just how they looked. But that's not really how it's supposed to be. The nomenclature is supposed to reflect how things are actually related. And so now that we're able to sequence everything so inexpensively, we can see, have a really easy insight into how everything is actually related. So I think all these changes are good because now the names reflect what they actually are, instead of reflecting what we thought they looked like 100 years ago. Yeah, I like them.
Yeah, I agree. I think it is a good thing, and it just kind of it just provides a little more clarity on the diversity of of life on this planet, and shows those evolutionary relationships, which I always found kind of fascinating, like how well. I mean, there's been some changes in the plant world, but like how consistent it was. Like it was actually a pretty useful system, and obviously it still is, because we teach lots of people how to do basic identifying off those same techniques that people have been using for forever.
Yeah, and one of the most fun things is to go out into the woods and collect a mushroom. I bring it home and sequence it, and then once you get that sequence data back, if it's something rare, you're the first person ever to see what the close relatives are to this mushroom. And half the time, the books are completely wrong as to where it actually fits in. Um, and then you can like build a phylogenetic tree and see how everything is related to it. And then when you see it again in nature, you have a much deeper understanding of how it got there and what it really is. So if you like combine like microscopy with the DNA sequences and taste and photography, you combine that all together, you get a real deep understanding of these organisms, and that's one of the funnest things.
Yeah, trying to see some of these reactions. The person who asked the question is excited about your answer. They just put a bunch of exclamation points. And I like, and I think those revisions in the nomenclature reflecting that deeper evolutionary tradition yeah, they said kinship relationships. I think that is important for highlighting that, because like one of the biggest things I try and help people understand about evolution is like there's no end goal in mind for evolution. Everything is just kind of evolving outward through time, and nothing is more evolved than anything else. So like we're really, when we learn about these deep genetic relationships, we're like learning about like different aspects of our, you know, ancestry on this planet from like on a very deep level, but very like non I don't know, woo woo level. Like it's ultra real. That's what I like about it.
Yeah, it's really neat. You know, another really cool thing is that anybody can go to GenBank or Mushroom Observer, iNaturalist, and download a lot of these DNA sequences. And even without having to sequence anything, you can figure out a lot of stuff that's not been published yet.
Very cool. Yeah, I hadn't even thought about that. Let's see. Have you heard of or tried Min ion portable DNA or RNA sequencer? I think the Min ION is like a brand name or something.
Yeah, maybe they call them Minion. I don't know.
Oh
But
yeah.
you've heard of them, and I'll probably be getting one within the next couple months. So that'll be a fun thing to try. And it'll be real interesting to see how good it is for what I'm doing. You know, the sequencing I'm doing is not usually full genome sequencing. It's more like just a little bit of junk DNA from each collection. But then MinION might be able to do that, like pulling a strand of DNA, and if it's not the one that I want, then reverse the polarity and kick it back out. So it'd be interesting to see if I can use that you know, in a cost-effective way for DNA sequencing the barcode genes, because usually people use them for full genome sequencing. And it costs about $1,000 per run. So that's pretty good price for a full genome sequence, but then again, that's just one sequence, and it would give you a whole hard drive full of data, which may or may not be useful. But it might be the future. You know, for right now, I think Sanger sequencing does what I want a little bit better. But I'll get one pretty soon and see what I think of it.
Let's see. Are there any psilocybe species that are fluorescent? So
No, all the psilocybe species are kind of dull brown, you know, brownish in color, and with the black light, they just look like shadows. They don't really convert any of the ultraviolet into visible light.
someone wants you to do
Diamond
another
mushrooms that are
poster.
fluorescent, like there's the Gymnopolis. You know, those have some fluorescent chemicals in them that have psilocybin as well.
But it's not the psilocybin that's making the fluorescence, is it?
Yeah, not the tryptamines itself.
Someone wants you to do another psychedelic mushroom poster from Mexico.
Yeah, that'd be a good idea.
Do you have any more trips planned down there for now, or is it all on hold because of the?
I don't have anything
pandemic?
planned right now because who knows how long COVID will last. Probably depends on how many people take the vaccine. Um, but as soon as, you know, I'll lead some more trips as soon as I can get down there and, you know, do it vaguely safely.
Yeah, that's understandable.
I know some people in Mexico say that Mexico is being hit pretty hard with COVID right now. You know, the hospitals are overflowing and stuff like that. So kind of like this country.
Yeah, it's pretty brutal. Let's see. I'm not seeing any.
I want
more
to chime in if you guys don't mind, Alan. I just have a quick question. Our group has briefly talked about doing a mobile lab in order to make it more accessible for us. And I'm wondering if you have any critiques or recommendations about how to make that successful.
Yeah, I think you definitely could do a mobile lab. And that's an interesting idea because then you could just like drive it over to someone's house and keep it there for a while and, you know, move it around. I don't think there's any reason that wouldn't really work. You know, you could have the microscopes and an autoclave and you know, DNA sequencing setup, and all of that stuff is perfectly mobile, as in you can just throw it into a car. So there's no reason you couldn't outfit a trailer to make it work, you know, pretty well and have it at like an official lab. So yeah, I think it's a pretty good idea.
Let's see. How did you get interested in mycology, and what's your educational background and history?
Let's see, my educational background, I went to high school in Illinois, and I got expelled from high school for sending thousands of pages of faxes to the school fax machine from a payphone. And then I never went to college. I just learned everything on my own. And I got interested in mycology about 2001 when I went hiking on Christmas Day down in Redwood City, California. And there was just mushrooms everywhere because Christmas is the peak of the California mushroom season. And I wanted to know like which ones were edible, which ones are poisonous, which ones were psychoactive, which ones were common and rare. And I figured there must be people out there that knew all of these mushrooms, but I didn't know how to find them. So I just started taking pictures of them and studying, and I learned a lot by editing Wikipedia. So that is, you know, that's how I got interested in it. And, you know, I was pretty bad at it until about 2007 when I started spending a little bit more time on it. And you know, about 2012 is when I quit my office jobs and just decided that it'd be a good idea to do it full time. It'd be a better use of my time than, you know, sitting in an office.
I always love hearing the story about how you have your last name. I know a lot of people are always thrown off by your last name. Do you mind sharing that story, or is that another?
Yeah, I was. It used to be that the Drug Enforcement Administration was raiding medical marijuana patients in California, and I didn't think that was a very good use of taxpayer money. So every time they would raid a sick person and bust them for weed, I would gather with a bunch of other people and we'd go picket the DEA office in San Jose. And that actually worked. After 3 different protests, they stopped doing that. But we would picket them with signs and the media would show up. And at the time, I was working for the federal government. So a reporter came up to me and she asked me why we were there, if I wanted to talk to about, you know, about the protest and what we were trying to accomplish. And I said, sure. And she says, what's your name? And I said, Alan Rockefeller. And because I didn't want to say my real name because I was working for the government. And they have crazy background checks and stuff. And it was going to be on the news. And then she said, is that your real name? And I said, yeah. And then she asked me about, you know, the protest. And I told her that I didn't think it was a good idea to spend our tax dollars busting sick people and all that kind of stuff. And she went ahead and put me on the news. And it was really good that I used the fake name because when they did all of the security clearance check stuff, they didn't know anything about that protest or anything about mushrooms at all. So it turns out that using a different name is enough to fool the federal government.
Good to know. Good to know.
Yeah, we know how that goes. They just let, with all the surveillance and technology that we have.
Yeah, I mean, you know, they did a really extensive background check on me and they found a lot of stuff, but it was like basically everything I had ever posted to the internet under my real name and everything I had ever said to a police officer every time I ever been pulled over. But, you know, they don't know what they don't know, and there's a lot they don't know.
Yes. Just go on Mushroom Observer.
Do you feel like you need a degree in mycology to be taken seriously, or is it just experience and just
It
accumulating
really depends on
knowledge?
what you want to do. You know, if you want to get a job as a researcher at a university, you know, those people will not hire people without degrees. Um, but there's lots of jobs in mycology that don't require degrees, and I don't think anyone really cares. You know, like the other mycologists don't really care if you have a degree or not, because, you know, getting a degree, you know, very often just means that you partied for a few years or your parents had money. You know, it doesn't really say anything about your ability to learn or anything like that. So yeah, it really depends on what you're trying to do. But certainly, if you're trying to publish new species or, you know, get a job at somewhere other than a university, you don't need a degree. But you know, every employer feels differently about that kind of stuff. You know, a lot of employers think that not having a degree is cool because that shows that you actually can learn stuff on your own, and you know, they tend to hire people like that. And other employers think that you know, it's really important because if you have a degree, it means you have rich parents, and so they want to hire those kind of people. It really depends. Um, depends. Um, I see one here. Any advice on making money from mycology and botany? And you know, certainly selling photographs. If you can take really good photographs and sell them to people that publish books, that's one way to do it. Forays is another way to do it. Or you know, if you have like a PCR set up and do DNA sequencing, there's people out there that want to give money to get their mushrooms sequenced, and like right now they have to mail them to Spain, and so there's certainly a good market opportunity for someone that wants to sequence the DNA of mushrooms and, you know, just do that. One could also write books, or, you know, there's a whole lot of psychedelics companies all of a sudden. So, you know, these psychedelics companies definitely need mycologists. So there's probably more opportunities now than there ever have been before. And some people even just use like crowdfunding, like Patreon, to fund research, or GoFundMe. Another option might be writing grants.
Someone was asking about the name and the Pacific Northwest mushroom that smells like burnt rubber. If you could repeat
Oh, that's
that one.
Rhodophyna nidalina.
Rhodophynin nidalina.
Rhodophyna nidalina. Yeah.
Okay, cool. Cool. Do we have any more questions out there on YouTube? We have to give them a few more minutes, a few seconds to catch up because they're a little bit behind us. But any more questions out there on YouTube and Facebook and in the Zoom chat here? If not, maybe we'll wrap this up. Let's see. When you, I guess this person commented on YouTube, said you can do your own research and sometimes even partner with someone at an institution for the research. That's kind of what you do, right? Like you got your papers published. That one paper that you showed us earlier, that was published like in conjunction with like a university.
Yeah.
Or?
I mean, it's not, I wouldn't say it's really partnering with them exactly, but sort of. Like one thing I did to learn DNA sequencing was just to volunteer over at UC Berkeley, and just you know, to help out in the lab and that sort of thing, and that works, yeah. But you don't really need anything about universities to get your paper published. You know, like the companies that run the journals, they don't check credentials or anything like that. But you know, and you really don't need it for DNA sequencing anymore. But you know, it's more like it's just kind of random that my co-authors, some of them happen to have jobs.
Word. What's your favorite place to hunt for mushrooms?
You know, it really depends on what time of year it is, because there's nowhere that's really good all year round. So I'd say like December, it's California's the best. November, Northern California. October, Oregon and Washington. September, Washington, British Columbia. And then before that, like the summer, it's really good all over the East Coast. So, everywhere from, New York to Florida, the Midwest, all of those places are really good. Michigan was really good. That's somewhere that I spent spent a couple months in Michigan this year, both in the lower part and then the upper part. There was a lot of really good stuff in Michigan. Vermont had really cool mushrooms. Florida and Eastern Texas have really cool mushrooms. I think some of the coolest mushrooms I have seen have been in Ecuador and Mexico. Southern Mexico has really cool mushrooms. Northwestern Mexico has really cool mushrooms if it rains, like Baja California. So, I mean, probably the best thing to do is just to look at a map of the rain and find out like where on Earth has had the most rain in the past month, and then just go there. And it'll be the best mushrooms. It'll be really good mushroom season. And just like look on Google Maps for like the largest trees or the greenest forests to find spots.
Let's see. Are there any particular mycena in the Pacific Northwest that bioluminesce?
Let's see, in the Pacific Northwest, yes, but most of them are very dim. Like Mycena hematopus. I found maybe like Mycena pura is one that's all over the West Coast, and the stem bases of Mycena pura glow. Mycena maculata is pretty common in the Pacific Northwest. The stem bases of that one will glow. There was only one Mycena that I found on the West Coast that glowed very brightly, and that was in California. There was one in Oregon that was sort of bright, reasonably bright. But most of the really bright mycenas are in Mexico and South America. There's a few in Florida. There's probably some in Texas and Tennessee.
Cool.
I see something about Rhabdomyces. I found that to be really dim sometimes and pretty bright other times. Like Rhabdomyces viridis, when it colonizes a whole leaf, the leaf glows pretty brightly, but if it's just a stick with bark, it's really dim. So kind of variable.
Do you have a favorite edible mushroom?
Amanita villosa is definitely the world's best edible mushroom. But that's only grows in California and a little bit into Oregon and Baja California. But, that's an amanita that comes up in the spring. It kind of tastes like scallops. So that one's really good. There's some from the Amanita rubescens group in Mexico that don't have names yet. They're probably in Texas and some other places too. A lot of the amanitas are really good. I like a lot of like the boletes, porcini type things. They're pretty good, but really any mushroom is really good if you're a good cook. And even the best mushrooms are pretty yucky if you're a bad cook. So the skill of the chef matters much more than the species of mushroom as to how good a mushroom is to eat.
I think that's a really good point right there. With the changes in nomenclature and taxonomy that you talked about earlier, how does that affect like field guides and such?
Yeah, I mean, the field guides are definitely outdated, and it keeps changing quite a bit. So I think it'll settle down in a few years, a little bit, at least for the bigger things. But yeah, for right now, the field guides are all out of date. But that's okay, because when a name is published, it remains a valid name forever. Just because a better name comes along doesn't make the old name wrong. So you can still use the old names as long as people know what you're talking about. They work pretty good. But it's kind of nice. It's cool to use the new names just because then the name you're using reflects what it's actually related to, but not super important.
Yeah, that makes sense to me. And like the mushrooms themselves didn't change. It's just how we're thinking about them and trying to talk about them, especially in that very rigorous academic sense. Like if you're just out there trying to have fun, as long as you can do it safely, then like I personally don't think it needs to affect your life that much. And let's see, wants to know, like, what was your transition like from your office job to full-time mycologist?
Like I would just, in 2007, 2008, I would sneak out of the office a lot to do mushroom hunting. I'd go on lots of runs and on all my breaks and look for mushrooms when I was out there. And spend a lot of time on Mushroom Observer, on iNaturalist at work, identifying stuff. And then, like around 2010, I started to quit my full-time jobs and just like spend 2 or 3 months in Mexico hunting mushrooms, and then get consulting jobs in the off time. And then, around 2012, I just decided that the computers were not really a good use of my time, and that I should try to just do mycology full time. And so that's what I did. And my boss was pretty surprised when I told him I was going to quit and to go mushroom hunting. He had never heard of that reason for quitting before, but they were like, okay, and never went back.
Hell yeah, that's sounds like a great transition. Okay, this one's gonna be the last question because I gotta get up and I have to go to work tomorrow. Do you know anyone who cultures brittle gill or milky cap and infects them with hypomyces to cultivate lobster mushrooms?
No, I've never heard of anyone doing that. I think it'd be pretty definitely possible and not too hard. Whereas you just take the mushrooms and throw them under the correct tree. Especially like if there's a bunch of russulas of the correct type, and then you could just like pulverize a bunch of lobster mushrooms and kind of throw them in that same little area. Then you can definitely start patches of lobster mushrooms that way. But I haven't seen anyone culturing them like that. I also bet you could culture the hypomyces just on agar without the russula. So I've grown hypomyces chrysospermis on agar, and that's the one that turns the boletes bright yellow. And it worked just fine. It turned the agar bright yellow, and it didn't seem to care that it was an agar dish. And there was malt extract agar. It didn't seem very picky about agar. I don't think those hypomyces are very picky at all. As long as there's some sugar there, they'll eat it. They're more picky in the wild, but it's in culture they're not very picky.
Very interesting. Well, thank you so much, Alan, for coming in and talking with us tonight. I'm gonna turn off the broadcast here, and we can stick around here for a second in our Zoom chat for just a little bit. But yeah, if anybody enjoyed what we put on here tonight, you can go over to centraltexasmycology.org and either give us a donation or become a member, and you'll help support more events like this in the future. And whenever COVID's over, we'll be doing lots more live events, and so it's a great way to get plugged into the Texas mycological community. So yeah, thanks for tuning in, and we'll see you next time.
Thank you.